right, this is a series of lectures that have to do with Mendelian genetics, classic starter genetics. Both this set of lectures, which will talk about monohybrid crosses, and in the next set where we'll talk about dihybrid crosses. These are absolutely critical things. I know you've been through it. You probably recognize, hopefully you'll recognize all the crosses, but people go, oh, I know that. I guarantee you, after many, many years of teaching this course, you don't know it the way you need to, okay? And I say that just for your protection, because it turns out that if you understand this part, if you understand monohybrid crosses, what we're going to call Mendel's Big Six, and then dihybrid crosses, which is the next series of lectures, once you understand that, then working these other problems, we're just going to layer on a little bit, okay? If you don't understand it, it doesn't matter how much we layer on, you can't grasp the other problems. So I really encourage you to spend a little time. This is the chapter in your book that deals with simple Mendelian. I would really encourage you to spend some time working the problems at the end of that chapter. Make sure you understand it, because one of the biggest things I have is we get toward an exam where we've got more complicated types of Mendelian problems, and students will come in and they're going, I just can't do this. No, oh, can you do this? They can do that part, and then I say, well, but wait, what about the simple part underneath? And they just go lost. Well, that's where they're to grasp it. So I really want you to kind of pay attention to these next series of lectures and really work some problems till you're very comfortable doing it. They're very simple problems. It's not difficult, but if you don't grasp it, it can make the others very difficult. Okay, now, this series of lectures, we're going to cover some terms. We'll talk about genes and alleles and loci. We're going to talk about homozygotes and heterozygotes. We've already talked about phenotype and genotype, so you should know that. We're going to introduce this concept of a mode of inheritance, is what we're going to call it. And we're going to talk specifically today about the principle of segregation, and talk about the big six, as we'll call them, the big six crosses that we're going to make. Okay? All right, so we have been often molecular because we feel like we kind of need to give you that background on what DNA is, a little bit about replication and things, so you understand a little bit about the underneath part. Now we're going to move into transmission genetics. Okay, and don't forget that there's a couple other areas we'll be heading into, such as cytological genetics and then eventually population genetics. But for now, we're going to move over into transmission genetics, and then we will loop back to molecular as we talk about, okay, now that you've got this genetic information, how do you get it converted over to phenotype? Right now, we're more talking about how do you transmit it, what are the rules, and then what happens once that occurs, right? So I like to use art and when I talk about these things. So here's a sort of an art picture, but where we've been up to this point, as far as domestication and all that sort of things, as we work through genetics, thinking about genetics and theories, is this kind of these flat profile pictures, right? Well, now in the 1800s, the middle to late 1800s, we really changed everything and brought forth this whole idea of transmission genetics, just like the old great Dutch masters, who added perspective. So they took these things, instead of being a flat picture, they allowed it to perspect back, give a three-dimensional image. They also added in things like shadowing to give contrast to it. Well, that's exactly what's happening here. We're going to take some basic genetics, and we're going to add a little bit of contrast. We're going to add a little bit of depth to what we have. And there's a person responsible for this, is Gregor Mendel. And Gregor Mendel was an individual that was born in what's now Czechoslovakia, was then at the time was Austria. He was from a very small rural family, at least in a small farm, and he did manage, because of support for other people, to go to secondary school, and eventually he made to the University of Worms. At the university, he took on the robes, so he moved into a monastery, so he became a monk. And this monastery wanted him to be a teacher, so they sent him out to the University of Vienna, where he gained his skills as a teacher. And in fact, he taught for a number of years in the system, because at the time, monasteries were one of the places of learning in the old world. Eventually, he became an abbot for the monastery and took on roles of being sort of the executive officer for it. Now, while he was teaching and doing other things, he really became interested, becoming from this rural background, about what can I, I want to do some research. I want to do some science. He actually started when he was working at the monastery with mice, breeding mice, looking for genetic traits that might answer some questions. And uh, the priors and others people at the monasteries kind of felt, well, breeding mice, that's kind of, there's some sex going on here, and they're mammals. That's probably not the best thing to have in a room somewhere in a monastery. So they've said, no, you can't do that. You need to find something else. So he said, well, I'll go out to my garden, which is a picture of its actual garden here, and peas would work. So he started taking peas. Now peas turned out to be pretty useful because you got a lot more progeny than you did with mice. Plus, you could hand manipulate them. You could take this one and hand pollinate that one over there, so you were assured of who got pollinated by what and the answers. Now, the big question he was after, we go back to our earlier discussion about history. He was after this concept of blending inheritance, right? This idea at the time that if we took two different things and we put them together, and we would get some blend of what those were. And that was the prevailing view. The seed salesman would come in and show you pictures of drawings of this and this. We put those together, and the seeds we're selling you should come out and have both traits or something. So he was wondering whether this was correct. And again, this was Darwin fought with the same issue, right? Because the problem of you get a large thing and a small thing, you always keep going back to the average if blending occurs. So he was really testing this concept of blending inheritance. Well, he worked with a number of varietals. In fact, he took peas and he started playing with them and taking very nice records and ideas, writing it all down about what he saw. Eventually, I think he had 34 different varieties that he looked at. It turned out that about 7 of them really were things he could kind of understand. We'll talk later lectures about why a couple of them were a little bit too difficult for him, but there were 7 that made some sense. They had sort of 2 contrasting results, a smooth and a wrinkled, or a yellow and a green, or whatever, and he was able to take these and look at them, and they all sort of gave the same answer when he did his crosses. So he selected these and did further work with these. Okay? Now, what he set up was what's known as a monohybrid cross. Okay? Initially for us, a monohybrid means one trait. Okay? So we take one trait. It may have contrasting phenotypes like tall and short or wrinkled and smooth, but he basically set these up. Again, he's trying to test for blending inheritance. And so he would start out with a pure system. We'll talk more about why we know these were pure, but he would talk out with pure parents. We call these the P1 or parental generation. We'll take some green peas and we'll say that, as he did, we'd cross them with maybe some yellow peas. And these were both parents. These were parentals, okay? Now, from these, he bred them together. He hand pollinated, and he ended up producing what's known as a first filial generation. We'll call that the F1. Filial is progeny generation. And the idea was, if it's blending inheritance, what he ought to get was something that looked something like one of these, right? He might get something that's got stripes in it, or partial blopsy yellows or whatever, or maybe something that's sort of in between in color, a light green or something. He was expecting to see something. He was expecting to see both the green and the yellow somehow coming out in that F1. Because that's what blending would do, right? It would blend those two colors together in some way, either as both solid colors being there as, polka dots or something, or just a lightening of the green toward the yellow range. When he did his crosses, he didn't get that. He got all green, exactly green like the green parent. No hint of yellow, no stripes, no dots or anything else. And, I think most of us would have said, well, there's something just odd here, and eating the peas and going on. He was a little smarter. He said, I'm going to take those peas, those green ones, and I'm going to very carefully take those F1s and I'm going to cross them amongst each other, okay? I'm going to come along and I'm going to take the F1s and I'm going to cross them with other F1s that I have from the same parents, and I'm going to produce an F2 generation. In other words, these are grandpods of the original parents, but this is the second filial generation. And let's see what happens there. Kind of a very interesting question, right? Do they all stay green? Do finally I get my blotchy yellows? What happens? Maybe I just go to the F2 and it'll show up. So when he did that, lo and behold, what he got were some perfect green ones, exactly like that grandparent plant that was green, and like those plants, the parent plants in the F1, and suddenly the yellow showed back up. Okay? And not only did this yellow show back up, but it showed back up in a very specific ratio across all 7 traits. It showed up in a ratio of about 3 to 1. Okay, so whatever yellow had disappeared in the F1, and it reappears in the F2 and part of a ratio of 3 to 1. Same thing happened for all the traits. One of the phenotypes, whether it was wrinkled or smooth or whatever, would disappear, and then it would all of a sudden show back up in the F2. Now, most of us would sit around and just scratch our heads. I certainly would have, just this information. But he's sitting there in this monastery. He's got time to think. And basically, if there had been Nobel Prizes at the time, he would have won one for what he came up with. He said, okay, I understand what's going on here. Those 3 to 1 ratios, those disappearances tell me lots of things have happened, and I understand what those things are. Okay? Now, what did he say? He said, I've got this way these things look. We call that the phenotype. I've got a ratio of that phenotype, which is 3 to 1, right? All right. Underneath that, there must be some other kind of ratio. In other words, the genetic things that are being passed. Now, remember, we're thinking of particulate passing at the time, right? Sort of these pan-genes idea. It doesn't really matter. He was just thinking underneath, there's a genetic ratio, and that genetic ratio is actually different. It's not a 3 to 1. It's, in fact, a 1 to 2 to 1 ratio. And what has happened here is that we have taken one of these, he called them unit factors, okay, one of these alleles, we'll call them now, we'll call them big G in this case or whatever. And when big G's there, it seems to take over and do something. And when it does, so if you've got 2 big G's, you're green. Even if you got 1 big G, you turn out to be green, right? Only way you can be yellow is to have completely everything else that must be there. So he figured this out, okay? And what he figured out was the genotypic ratio gives rise to the phenotypic ratio, okay? The genotypic ratio is determined by whatever cross we make, what we pick as parents, right? A green one and a yellow one, or wrinkled and smooth, or 2 smooths, or whatever. That determines ultimately our genotypic ratio. And then what determines our phenotypic ratio is a modification of that. We're going to call that modification, that thing that happens, the mode of inheritance, okay? So he said, there's a mode of inheritance here. I know underneath I've got this repeating genetic issue, repeating outcome, but what they're showing up as phenotypically is by this other rule, which we're going to call the mode of inheritance, right? So in the next lectures, we'll talk about these modes of inheritance, and we'll fill in some of the genotypes that belong here as we work through it. And then after that, we'll actually go back and work some problems and see how that works.